Hi, I'm Pedro Correia. I'm a postdoc at the University of Copenhagen, and I'm working on this project called Deep Roots, related with wheatgrass domestication, along with another postdoc, Java Najafi, and we would like to thank the organizing committee to letting us presenting our work in this meeting. I think by now everyone knows why it's important to grow wheatgrass, and what are the challenges to increase the yields. So our main goal is to show how can we contribute to this goal. And so the main goal of our project is to identify wheatgrass genes that are related with domestication genes in other grain crops, so that those wheatgrass genes can be targeted by mutagenesis or genome editing approaches, helping to accelerate the domestication of wheatgrass. In our study, we are using by now barley as a model because barley is a diploid. We know a lot about the barley domestication process. We have strain-forward protocols for transformation and mutant regeneration. And we also know from previous studies that there is a good similarity between the barley genome and the 3 subgenomes of wheatgrass. So just to better visualize our approach, we have a domesticated barley, and if by expressing wheatgrass genes, we can revert the domesticated phenotype to a wild type, we can prove that these wheatgrass genes are good targets. By now, we are working with 2 different genes, Brittle rachis 1 or BTR1, more related with seed dispersion, and dense and erect Panicle 1 or deep 1, more related with plant architecture. So for BTR1, we know that our domesticated barley carries a mutation on BTR1, so it has a domesticated phenotype where the seeds stay attached to the spike, so that we can harvest them easily. And if by expressing putative BTR1 wheatgrass genes, we can revert the phenotype to a phenotype where we get more dispersion of the seeds, we can prove that this gene is a good target. We also know that in Golden Promise, the mutation leads to a truncated protein, and we could identify 6 different isoforms of wheatgrass related with BTR1 and barley. So our strategy is to clone the coding sequencing in the sequence in an overexpression vector with a strong constitutive promoter, and also to clone the promoter with the coding sequence, leading to the complementation in barley. By now, we clone and transform 2 wheatgrass isoforms in barley. In isoform 1, we are now regenerating mutant plants, and the other, we are still in an early phase. The other gene is dense and erect Panicle 1, deep 1, and so in our greenhouse, we are growing Golden Promise along with wheatgrass, and we can see that our barley has a compact phenotype, and wheatgrass is almost leaving the greenhouse. So it would be interesting to get wheatgrass with a more compact phenotype so that we can harvest the seeds easily, and it will be also better in climates as in Denmark, where very windy, and so we get lodging resistance. So we know that the mutation on deep 1 can lead to a more compact phenotype in rice and barley, and deep 1 encodes a heterodynamic G protein, gamma subunity, and gamma and G proteins are related with development process in animals and plants. In Golden Promise, we know that the mutation is this zone, and also in rice and barley mutations on this cysteine-rich protein domain can lead to changes in the plant architecture. So if we express wheatgrass genes and get more wheatgrass-like phenotype, we can prove that this gene is a good target. By now, we could clone 2 different isoforms, and we are now expressing it in barley, and we are regenerating mutant plants. Our next targets would be genes related with spike density, grain size, vernalization requirement, and photoperiod insensitivity. And just to finish, I would like to thank our PI, Michael Palmgreen, and our collaborators, Guanmi Lu and Gachagao Group in China, and the Carlsberg Wheatgrass team that is collaborating with us in this project, and also our funding partners, Novo Nordisk Foundation through the Novo Crops project, and Innovation Fund Denmark that is funding our project. So thank you. Questions and suggestions are more than welcome.